Hi everyone, welcome back. So our today's topic is gel electrophoresis. So you are learning gel electrophoresis as a part of separation, as a separation technique. Okay, gel electrophoresis. You are learning as a separation technique. What is gel electrophoresis? You know, you have studied the action of restriction endonuclease enzyme. Okay, the restriction enzymes, especially the restriction endonuclease enzyme, can cut the DNA at various defined sites. You have learned that wherever the palindrome or the recognition site is there all this restriction enzyme can cut the DNA. When it makes cut randomly, it is surely producing random fragments. The DNA is cut into many random fragments. Then how is it possible? Manually, how is it possible to pick out the DNA which we need? The desired DNA, can we pick out from this? No. So how is this possible to separate the DNA from a cluster of random sized fragments? So after the action of restriction enzymes, we have a lot of differently sized DNA fragments. How can we separate it and how can we isolate the DNA segment we require? That is by gel electrophoresis. What is the technique? So we can say this is a separation technique. The technique helps to separate DNA fragments, and also we can say this technique helps us to isolate our desired DNA based on the size. When it is separated, we can take the desired DNA what we need. Okay, and what is done in this technique? In the gel electrophoresis setup, when we draw it this way, see, on a medium, on a platform, gel is taken. So gel is taken in a platform. I'm just giving a 3D view here. Whatever is it. Okay. Okay, whatever is the outline. See, at one end of this, there are certain notches, or we can call them as wells. Here I am drawing 4 wells only. These are named as wells. These are the wells. Okay. Wells. See, and this is the setup. Here the full setup, the matrix is agarose gel. Most commonly used gel is agarose gel, which is obtained from seaweeds. Agar, you know, gelidium. Okay. Then anyway, what is taken in the as matrix here is agarose gel. Also polyacrylamide gel electrophoresis is the page. Instead of agarose, polyacrylamide gel. The other gels are also used. The most commonly used gel is agarose gel. Okay. Then listen to here. See, this setup when is connected to power. That means under an electric field. Okay. Under an electric field. Suppose a battery or any connection, you just imagine. No need to draw it. Okay. Suppose this side is the cathode and this side is the anode. Okay. No need to represent it in the diagram. Just we are taking the DNA fragments and introducing into this well. Okay. The DNA fragments after the action of restriction enzymes, we are introducing the fragments as such as sample. We are loading to this wells. Sample 1, 2, 3, 4. We can load into this wells. Okay. So the DNA samples we can load. Then what is next is under an electric field, when a power is given, under electric field, this DNAs will move towards the anode. Okay, you know DNAs are here. There are number of fragments of DNA. You know the DNAs are negatively charged. Why the DNA samples are negatively charged? You know that every nucleotide of DNA consists of phosphate. Phosphate is giving, phosphoric acid is giving negative charge to DNA. This all facts we have discussed. Okay, DNA samples will be added into this. Okay, what happened to the DNA fragments under an electric field? This DNA fragments will move, okay, in such a way that larger sized fragments will move slowly, okay, whereas smaller sized fragments can move fast. This way it gets separated based on its size. Okay, DNA fragments can be separated based on its size. Okay, if it's undigested, not yet digested, it will be remaining like this. Okay, anyway, according to the size, they are separated. Why? The DNA fragments move in such a way that larger sized fragments move very slow, smaller sized fragments can move fast. Okay, will move towards the anode, and this type of movement will separate the DNA according to their size. Large size will remain to this side and the smaller sized fragments will remain to that side towards the anode. Okay, so this is a separation technique. Then tell me which are the major principles, on which principle, on basis of which principle it works. Why is it possible for the DNA fragments to be separated? The first case is since DNA shows charge. Okay, if the DNA does not, fragments does not show any charge, we cannot separate by this method. Okay, based on the charge they separate. Because of the charge they are moved to anodes. Okay, since they are having negative charge, they are moved to anode. And the second important thing, imagine instead of gel we have a platform, a thin, okay, anything, a tin or anything there. There is a hard background. It cannot move. And second important principle is the sieving efficiency of the gel. Okay, the DNA is allowed to move, allowed to hold in the sieve and allowed to move because of the sieving effect of the gel. These two are the principles of gel electrophoresis. The DNA molecules are able to separate, the fragments are able to separate because of the presence of the charge, okay, under an electric field and also due to the sieving effect of the gel. Okay, simple fact. Then, so this way the DNA is separated. This way the DNA is separated. Then what is next? We cannot see this DNA like, see the DNA we cannot see in any color. So what we should do is, we should do staining process to visualize this. In order to observe the DNA, we have to follow the staining process. Here we are using ethidium bromide as the stain. Okay, DNA can be visualized using ethidium bromide and it should be followed by UV illumination. Then we can see DNA as dark orange bands. Okay, see when staining is done with ethidium bromide followed by UV illumination, this all DNA fragments can be visualized as dark orange bands. Okay, then after this there is a step associated with this known as elution. Okay, the step associated with gel electrophoresis is elution. In this step the DNA fragments can be cut out. The process of cutting out of the DNA fragments from the gel is known as elution. The process by which DNA fragments are cut out or removed, cutting out from the gel is known as elution. So that is about the gel electrophoresis. It's a technique of separation or isolation of DNA fragments. This is possible because of the negative charge of the DNA, and under an electric field the DNA fragments are moved towards the anode and the larger fragments are moved slow, whereas the smaller fragments can move very fast. Okay, then another important thing, during this separation, the sieving effect of the gel makes a special role. Okay, so that's about the gel electrophoresis technique, and then study the staining using ethidium bromide and UV, and also study the elution process by which the DNA fragments can be cut out from the gel. So, dear students, you can see various setups for gel electrophoresis in this presentation, and listen to the bath where you can see the DNA fragments are separated according to their length. So you can see differently sized DNA fragments separated. And you have already studied this gel electrophoresis has a great application in DNA profiling or DNA fingerprinting. Okay, just listen to the different setups. That's all about the gel electrophoresis, and we will discuss another topic in our next class. Thank you.